This is the structure of the glomerulus, where there's really a very high pressure within the Bowman's capsule. So the blood enters the Bowman's capsule from the afferent arteriole. It's L, not D. And then there are three most important structures within the Bowman's capsule, which are podocyte, basement membrane, and fenestrated wall of capillary. So fenestrated wall of capillary has small gaps through which the fluid from the blood gets filtered out of the capillaries into the lumen of the Bowman's capsule. And then second structure is the podocyte. Podocyte has finger-like projections, which are called as podocyte foot processes, and they prevent blood cells from escaping the capillaries, the gap between the walls of the capillaries. The basement membrane acts as a barrier between the capillary walls so that they don't stick to each other. And then it also has negative charge, which repels most of the blood proteins, which also have negative charges. And then glomerular filtrate is the fluid which has been filtered through all these three structures from the lumen of the capillary into the lumen of the glomerulus.
The proximal convoluted tubule is a structure that's connected to the Bowman's capsule. It's a folded structure where selective reabsorption occurs, and it's the second stage of filtration in the kidneys. There are several characteristics of the proximal convoluted tubule that aid selective reabsorption. The single epithelium layer in the tubule minimizes distance that the reuptake materials have to travel. Second, tight junctions prevent passage of waste materials. Third, mitochondria allows active transport in the proximal convoluted tubules. And fourth, the microvilli increase surface area. All glucose and amino acids are reuptake by selective reabsorption, whereas 80% of water and ions are absorbed. Active transport aids the passage of materials past the apical membrane and basolateral membranes. The apical membrane is the membrane that's facing the lumen of the tube, whereas the basolateral membrane is the membrane on the outside of the tubule. Mineral ions in the filtrate are transported via protein pumps actively at the apical membrane. These include calcium ions and chloride ions. And sodium-potassium pumps that's located in the basolateral membrane aids the transport of sodium ions actively outside the basolateral membrane. However, the concentration outside the tube is high, whereas the concentration inside the epithelium cells are low, so sodium ions from the inside of the lumen move passively into the cells of the epithelium. All amino acids and glucose are co-transported via sodium symport pumps by the energy released from the passive diffusion of sodium ions, and vitamins are transported by carrier proteins. H2O is reabsorbed via osmosis because of high solid concentration in the epithelium layer.
So I'm now going to talk about the loop of Henle, which is the U-shaped structure that can be found in the medulla, which is the lower section of inside the kidney. So the loop of Henle, there's two things that we need to notice about this U-shaped structure. So first, it's in the medulla, and around this loop there is what we call the vasa recta, which means it's a network of capillaries and blood vessels that go all around the loop. And that makes it easier for our body to reabsorb the nutrients, electrolytes, and the water that comes from the filtrate that goes through the lumen of the loop. And then secondly, the loop can be separated into three different sections. So there is the descending loop of Henle, which is the initial down part of the loop, and the thin and the thick ascending loop of Henle. And the reason why we separate this U-shaped structure into three different sections is because all these sections have different adaptations for their functions. So what is the main job of the loop of Henle? So the main function is to actually create an electrochemical gradient in the tissue that surrounds the loop, meaning that it creates a high solute, highly concentrated and hypertonic environment in the interstitium. The interstitium basically means the tissue that surrounds the loop. It's also called the interstitial fluid, and I'll just be using this term to make the explanation easier. Right, so first let me talk about the thick ascending loop of Henle, because this is where the concentration gradient is initially created, and this concentration gradient will eventually allow the descending loop of Henle to allow osmosis, so water movement from inside, from the filtrate to outside, and so the reabsorption of water from our body. Right. So in the thick ascending loop of Henle, the reason why it's called a thick ascending loop of Henle is only because the epithelial cells that go all around the membrane are thick, large. So I'll just show a really quick picture. So as you can see, this means that those thick and large epithelial cells are tightly packed together, which means that they have really tight junctions, and because water molecules usually move out through a membrane passively and through these junctions, because the junctions are so tightly packed together, water molecules cannot pass through this membrane. So that's why we say that the thick ascending loop of Henle is actually impermeable to water. There are two different types of transport that occurs in the epithelial cells. So first, there is active transport happening where sodium is pumped from the inside of the epithelial cell to the outside, to the interstitium or the surrounding tissue of the loop. So by doing so, it means that inside the cell there'll be a low concentration and outside a higher concentration. And that means that sodium from inside, other sodium ions from inside the filtrate will move passively down their concentration gradient from a region of high concentration to a low concentration. And because this happens through a co-transport protein, it will allow other ions such as chlorine and potassium ions to also move from inside the filtrate to outside of the loop. And so because of this movement of ions, it means that the interstitium or the surrounding tissue or interstitial fluid or the whole tissue of the medulla will be more hypertonic, more concentrated than the fluid inside the lumen of the loop. So now we'll go back to the descending loop of Henle, where osmosis actually occurs. So like we just said, the ions in the filtrate moved out to the interstitium, and so we can imagine that this whole region outside of the loop is really concentrated. So the water molecules that were initially coming from the Bowman's capsule, as we previously mentioned, all those water molecules, once they arrive at the descending loop of Henle, which is a permeable membrane, all those water molecules will move out. So by moving out of the filtrate, it means that the filtrate will become more concentrated as the water molecules move out. And those water molecules and nutrients will be reabsorbed by the body because of the vasa recta that surrounds the loop of Henle. So this whole process is actually called the countercurrent multiplier system. Countercurrent because the filtrate moves in opposite directions at different sections of the loop of Henle, so first descending and then ascending. And this opposite movement allows to create a highly concentrated region outside of the loop of Henle in the medulla. And so because of this highly concentrated region, water osmosis can occur at the descending loop of Henle, and water is reabsorbed by the body.
So once the filtrate moves past the loop of Henle, it enters the distal convoluted tubule and the collecting duct, which is where further reabsorption of water occurs. This is because the filtrate inside the tubules is hypotonic after reabsorption, whereas the conditions in the medulla is hypertonic. So water is drawn out of the tubules into the medulla, causing reabsorption of water. When the body is dehydrated, blood solid concentration is high. So osmoreceptors in the hypothalamus detect this, and it it stimulates the posterior pituitary gland to secrete antidiuretic hormone, ADH. And ADH is a hormone that increases the permeability of the distal convoluted tubule and collecting duct by increasing the number of aquaporins, which are channel proteins for water molecules. And so water is reabsorbed further, causing a small volume of concentrated urine to be produced. And so this lowers the blood solid concentration. Now, when the body is hydrated, the solid concentration in the blood is low. ADH is not secreted, and relatively little water is reabsorbed. So a high volume of urine, which is dilute, is produced, and the blood solid concentration is increased. So this process of balancing blood solid concentration and water levels in the kidneys is called osmoregulation.